Okay, so Gregor Nagel will be talking about matrix metalloproteinase-sensitive multistage nanogels. He is now a postdoc on Charité Hospital in Berlin. And now, Gregor, we have 10 minutes for your presentation.
Yes, welcome also from my side, and thanks for organizing this symposium. I would like to address some challenges that are faced by nanogels that are designed to deliver a drug to solid tumors. And I will start right with the challenges of these nanocarriers in the delivery of solid tumors. So the first one is the specificity of drug release. So what we want to have is, of course, a carrier that has a stable bond or connection to its cargo during the circulation time in the bloodstream, but then magically releases the drug in the malignant tissue. And for this, of course, the choice of release mechanism is crucial. And the second point I would like to discuss is that these nanocarriers that often take advantage of the EPR effect accumulate in the tumor tissue, and it's known that particles around 100 to 200 nanometers are especially prone to do that. But when they reach then solid tumor, they tend to get stuck close to the vasculature and does not really penetrate the tumor, and this poor tumor penetration is a huge problem for these kind of drugs because due to the poor vasculature and also to some interstitial pressure, we get a very heterogeneous distribution of these nanocarriers, and this of course leads to impaired treatment with these nanocarriers and can even lead to the development of drug resistance and the regeneration of malignancies because the tumor is not cured properly. So in order to address this, we need nanocarriers that are able to adapt to the barriers. And I would like to present our approach to this challenge, which is namely a multi-stage drug delivery system. So what we envisioned here is a peptide cross-linked nanogel in the size range of around 200 nanometer, which is able to accumulate in tumor tissue by the EPR effect. And then in the next stage, the nanogel can be degraded or fragmented by an abundant class of proteinases in the tumor microenvironment. So it's sensitive to an endogenous stimuli in the tumor microenvironment and is degraded to smaller particles in the size range of 30 to 50 nanometers. And these particles can then be better penetrate into the dense tissue and can be regarded as polymer drug conjugates because there's also anti-cancer drug conjugated covalently to these particles, which can then be taken up by tumor cells and release their cargo upon trigger. Next, I would like to show you the design and the synthesis of these particles. So they basically consist of two main building blocks. One is dendritic polyglycerol, which is functionalized with cycloactine groups, and then a fluorogenic peptide cross-linker, which has acyl groups in order to allow a cross-linking by strain-promoted cycloaddition. And in detail, this cross-linker consists of an amino acid sequence, which can be cleaved by this proteinases I talked about. And also the cleavage side of the linker is framed by a di pair, a Fred di pair, so which means that when the amino acid chain is intact, the fluorescence of one di is quenched, and upon cleavage, the fluorescence is regained of this di. And using nanoprecipitation, which was already explained by Pradeep, we obtain these nanogels. So this is basically that you have two solvents which are miscible, and but the polymer is only miscible with one of the phases, so it precipitates upon mixing, and this gives you very nice spherical particles. And post-synthetically, we also attached the anti-cancer drug doxorubicin using a derivative with a hydrogen bond, so that the doxorubicin is attached to the polymeric part of the nanogels in a covalent fashion and stable in physiological drug can be cleaved upon acidic pH. So by this, we obtained our peptide cross-linked nanogels, which also have an intrinsic reporter moiety, which can be used as a fluorogenic probe. So we played around a little bit with the degree of functionalization of the surface groups of the polymer, and also with the feed ratio of the cross-linker. And by this, we could tune the size of the particles and the cross-linking density between 200 and 700 nanometers. And in TEM, we see that we get spherical particles with quite low size distribution. And as I mentioned, with this intrinsic reporter unit, we can follow the degradation, because when we cleave these bonds, the fragments start to fluorescent. And we can nicely follow this over time with a plate reader setup. And we see that depending on the conditions that were used for the synthesis, that we get different kinetics for the degradation. And we see here in this direction that the degradation gets slower. And we can also correlate this fluorescent signal with the degradation of the particles. Here the particles were incubated with proteinases, and we also synthesized a non-degradable control, which was prepared with the same linker but with D amino acids. And we see that upon time and incubation with the enzymes, we see first an increase in size, which indicates a swelling of the particles, because the first cross points are cleaved, and then the size rapidly reduces over time to a fragment size of around 50 nanometers, which is exactly the size range that what we are targeting. But now we want to see if this size reduction of these particles also translates into enhanced penetration. And for this, we first labeled the nanogels on the polymeric part with size 3 dye. And we used multicellular tumor spheroids as an in vitro experiment. So these are small cell balls which are frequently used for penetration assays. And by optical sectioning in a fluorescence microscope, we were following the penetration. And for the free dye, it looks like something like this. So this is freely penetrating into these spheroids, whereas the non-degradable control is barely penetrating into these spheroids. And for the degradable nanogels, we see that it's not going as deep as the free dye, but apparently deeper than the non-degradable nanogels. And since we were not so convinced by the optical sectioning, because you cannot go in so much depth, we also prepared cryosection of the incubated spheroids and measure 2 different time points. So we see here after 2 hours, the free is already penetrating into the center of the spheroids, whereas the non-degradable one is barely visible. There's only a little bit on the surface. And also the degradable one is not really penetrated yet, but we see after 16 hours that also the degradable one is penetrating into the center of the spheroids, indicating that the fragments after degradation of the nanogels can freely penetrate into this tissue. This is even better visible in these magnifications of the spheroids. So we see here that the fluorescence of the degraded particles is penetrating deeply. So this means that the carrier is already the penetration of the carrier is already promoted. But now we need, of course, to check if also the cargo goes along with it. As a reminder, we had this intact nanogel, which is first degraded by MMPs to shrink in the tumor microenvironment, and then upon cell uptake, the drug is released in acidic cell compartments. And so here we followed conveniently the fluorescence of the doxorubicin as a fluorescent drug, which is also freely penetrating into the tissue and also is found then in the nucleus. Then from the non-degradable nanogels, we see again that there's barely any fluorescence of doxo, so it seems that the nanogels are not penetrating and are also not uptaken and are simply washed off the spheroids. And in the last image, we see that the degradable nanogels are penetrating quite deep into the tissue, and that the fluorescence is evenly distributed through the tissue. Now we also wanted to check, of course, the if the drug that is transported is still active. And here we see the cytotoxicity of the drug directly on the spheroids. So we measured the ATP release directly from the spheroids, and we see that the vehicle alone doesn't show any toxicity. And surprisingly, or not surprisingly, but luckily also the non-degradable nanogel doesn't show any toxicity, even though the drug is also cleavable on this part, but due to the fact that the nanogel is not penetrating and is simply washed up, we don't see any toxic effect. On the other hand, with the degradable nanogels, which are penetrating to the tissue and releasing the drug, there is an apparent effect on the viability of the cells. So with this, I would like to sum up the talk. So I showed you that the challenge of poor tumor penetration can be overcome by the synthesis or the design of a smart multistage system. We introduced here a protease-sensitive crosslinker to introduce controlled degradability, and the size reduction that was achieved by this promotes the drug penetration into the tissue model. And yeah, we could show that this carrier could deliver an active drug into the spheroid center. This work was recently published in Theranostics, and if you want to see more details, you can check the paper. And for this, I would thank you for your attention. And about myself, Marcelo already mentioned, I moved on to the institute at Charité. We're working now on a European project called Endoscape, which deals with gene therapy, of course, also with nanocarriers and nanoparticles. Thanks.
Thank you very much, Gregor. We have already two people that raised their hands. First is Facundo Matea from Córdoba, Argentina. Facundo.
Hi, Gregor. First, a very nice presentation and very nice to hear from you. It has been such a long time. And my question is about, do you think that these nanogels could be cleaved before getting to the tumor? Could you get a smaller size and losing the enhanced retention permeability effect before getting to the tumor or not? Is that possible?
So what I know is that these so this linker is quite specific for these matrix metalloproteinases, and they are not only found in tumor tissue, so they're overexpressed often in tumor tissue, but I think they are not found in the bloodstream. But when the particles end up in another tissue, it can happen that to a small degree or to some degree they're already degraded there. Yeah. But okay. Which is also a good thing, because then they do not get stuck in this kind of tissue and can be washed out, which is also a disadvantage of many other nanoparticles, that they accumulate and because they are not biodegradable.
Okay, thank you for the answer. Excellent.
Yeah, and of course,
next question is from. Sorry. Next question from Pradeep.
Yeah, nice, Gregor. Nice to hear you. So my question is more fundamental. Like, if you conjugate the doxorubicin beforehand and then you do nanoprecipitation, is it creating some problem? Because when you couple it later, it maybe they are more staying on the surface. Do you think it has make some effect?
Yeah, it was actually this was my first idea to have polymer drug conjugates and then cross-link them to nanogels. But there I had a lot of problems with the nanoprecipitation and with the stability of the particles. So I couldn't get stable particle, and I got always precipitation. So I think it would even better to somehow find a stable system where you first have the conjugation of doxorubicin, because it's also more controlled in the end. I think I even have when I do it post, I have a mixture of encapsulated doxo and of conjugated doxo, which I also see in the release profile. So I have in the beginning quite fast release. So for sure, from the design, it would be more clever to do this before, but I couldn't manage when I was trying that.
And I have another question. Did you compare also with some preparing particle around 40 to 50 nanometer, that how they behave? Because you are targeting, don't you think is 200 nanometer is too big?
Yeah, that is actually a control that is missing. Yeah, it's true. I should have.
Okay. Okay. Thank you.
Thank you for your input.
Okay, thank you, Gregor. Thank you very much. Thank you for the questions.